Okay, so we're small, but we're good. And plan today is to really discuss Salmonella. And what I hope to do is we'll start off a little bit, probably with many of you know, just sort of a overview of Salmonella and generally some of the dogma and just general knowledge we have on Salmonella. But then I want to sort of challenge things and shake it up a bit and sort of push the envelope with some of the things we found about Salmonella recently and how it all pertains to the whole infectious process, because there's been an awful lot of people who have studied Salmonella for an awful long time, and what you'll see is they really continue to find amazing things and really don't understand this organism all that well still. So obviously the concept is we'll talk about Salmonella and its interactions with the host, and one of the concepts I want to drive across is depending where the organism is, you really have different kinds of disease and different kinds of interactions with the host. So just to remind us again, Salmonella, it's a foodborne infection. Really, there's two flavors of infections in people. There's the systemic typhoid type fever that you see mainly in developing countries, and then there's the non-typhoidal Salmonellas, the gastroenteritis, nausea, vomiting, diarrhea that you see pretty much around the world, the food poisoning type Salmonella. And you can see the numbers here. Obviously, this still plays a major role in morbidity and mortality worldwide. And compounding this is the incredible levels of antibiotic and drug resistance in Salmonella throughout the world. So it's continues to remain a major problem in terms of foodborne infections. So Salmonella, as I said, is foodborne. You get it orally, ingest contaminated food or water, and then it goes down to the small bowel, intestine, where the bacterium then penetrates, probably through M cells and other cells, into the underlying phagocytic cells, macrophages, and it can cause intestinal problems, or it can then go on and distribute more systemically as it moves along. So we know that Salmonella has the capacity to penetrate the intestinal epithelium. There's been a lot of work done in this area over many labs over the years, interact with the immune system, both innate and acquired, and then it basically drains in the mesenteric lymph nodes, and there it can go systemic, even into the bone marrow, as well as the spleen and the liver, and it can then go down the liver into the gallbladder. We'll say more about this in this talk later. And then can then lead to reseeding of the organism in the feces, and then shedding, and then recontamination of other people. So Salmonella has historically been considered an intracellular organism. It has the capacity to enter into mammalian cells. So if you have the intestinal epithelium here, the bacterium can somehow interact. We really don't know a lot of this process yet. Trigger events in the mammalian cell, so it then extends these pseudopods up and around, embraces the bacterium, and then internalizes the bacterium into a membrane-bound vacuole. Now, some bacteria get in and break out of this vacuole, but Salmonella remains in this membrane-bound inclusion, where it can either transit through the cell or live in here and replicate, always remaining within this membrane-bound vacuole. So Salmonella targets phagocytic cells. This is an experiment we did many, many years ago where we wanted to see what kind of cells it was actually targeting the liver and the spleen. And basically it turns to macrophages and also neutrophils. And so the yellow is stained Salmonella, and you can see this heavy infiltrate of macrophages and then neutrophils to the site of infection. And there's actually a raging controversy in the literature for a long time whether Salmonella was truly an intracellular organism or whether it was just hanging out in the vicinity of these cells. And I think we sort of finally answered that with basically the invention of confocal microscopy. And we were then able to do cross-sections of in vivo infected cells with Salmonella. And the moral of this story is here's Salmonella in orange, the macrophage in green. No matter how you slice this thing, the bacteria is truly inside these cells. The reason the controversy exists is people had been doing electron microscopy, but because they had to use such high doses to see any bacteria, people were saying that was artificial. And with these high doses, there were a lot of bacteria outside the cell as well. So we really believe it does live inside cells, both phagocytic and non-phagocytic. So there's been an awful lot done on how Salmonella gets into cells and causes effect in cells. And I think we all generally know that that's due to these type 3 secretion systems. And these are specialized secretion systems that span the bacterial membrane that have the capacity to deliver molecules we call effectors into host cells. And these have been heavily studied in many gram-negative pathogens. And it really gives the bacteria a really nice mechanism to drive bacterial molecules into mammalian cells to reprogram it. Well, in the case of Salmonella, it's been well known for a long time now, there's actually two type 3 secretion systems. So it has two ways of getting bacterial proteins into cells. And generally, it's thought that this first one, called Salmonella pathogens island 1 or SPI-1, is involved in the uptake of the bacterium into the intestinal epithelium. So this is the initial stage of the event, where it can then pump molecules in that triggers basically invasion and entry into epithelial cells. This event also seems to deliver effectors that involve in the early events of gastrointestinal inflammation. So when we think of Salmonella and gastroenteritis, we generally think of this SPI-1 system. And the dogma in the field sort of suggests that this evolved first. It got the bacterium into cells, but then once you're inside cells, you have to figure out how you're going to survive inside these cells. And it then acquired a second type 3 system, this thing called the SPI-2 system. And like SPI-1, it delivers molecules, but different molecules in the mammalian cell. And work over the years in our lab, David Holden's lab, and many others have shown that this system seems to be involved in the bacteria surviving inside phagocytic cells, macrophages, etc. And it's thought to be involved in the systemic type of disease, so the later events seen in typhoid fever, etc. So the dogma says it's gastroenteritis for SPI-1 and then systemic diseases such as typhoid fever for SPI-2. So we've learned a lot over the years, and I'm not going to detail this because I think we're all pretty much aware of this, though, that Salmonella has this SPI-1 type system. SPI-1 basically delivers effectors that basically interface with the actin of the cytoskeleton and cause major cytoskeletal events, ultimately neutralize the bacterium and big actin of the cytoskeleton, rearranges to get the bacterium inside the cell. Once it's inside the cell, what Salmonella does is it basically disrupts the normal trafficking of the cell. So it basically blocks fusion with the lysosome. So you have a Salmonella-containing vacuole that goes in here. It has markers of the early endosome, but then it basically blocks the later events, the maturation, as it would normally, endosomes normally progress down this pathway. It blocks it. Now, remarkably, it also triggers these tubule structures here that called Salmonella-induced filaments, or SIFs, which we discovered many, many years ago now, and these are somehow involved in the bacteria replicating inside the cell. So the bottom line is it uses its SPI-2 systems to basically block the delivery to what would be normally toxic lysosomes to survive. And that's kind of reiterated here too, and as you can see, we know less about the SPI-2 system. You can see all the question marks here about some of the yellow Salmonella effectors and what they're doing, and really I think this is a more poorly understood type 3 effector system than the SPI-1 system, which Glenn and others have really worked out. So what we started to realize over the years is the SPI-2 system, their effectors play a key role in Salmonella inside these cells, and the molecule that triggers these Salmonella-induced filaments, these things, is an effector called CFA, and many of the SPI-2 effectors end up accumulating on the vacuolar membrane around Salmonella, where they seem to have effects, although others seem to be able to go distally and then trigger other effects in the target in these cells. So this is kind of an overview of what we know about the SPI-2 system, and the bottom line is it really does disrupt the programming of the cell. It goes to great lengths to really muck up many aspects of the vesicular trafficking system and even cytoskeleton involved in there. And many of the SPI-2 effectors have particular roles in affecting this inside cells. What I'd like to point out is that really most of this, and pretty much all of it, has been worked out in tissue culture cells, in vitro, as we call it here. And really how this pertains to the whole Salmonella biology, I think, is still really understudied. And that's sort of shown here. This is a list of known Salmonella type 3 SPI-2 effectors that we know are injected into mammalian cells, and we've been able to, well, not we, the field has been able to assign functions to, our lab's involved in several of these, but others not, to some of these things. But you can see there's several of these things we just don't have a clue what they're actually doing inside the host cell yet. Not only do we not know what they're not doing, we also think that there's probably other SPI-2 effectors that we don't know yet that might be involved in this process. So that led us on a hunt. So could we identify pretty much all the Salmonella SPI-2 effectors? So we knew what we had to deal with. And this is work that was done by Sigrid Ottweiler and Amit Bhavshar in the lab. This actually just came out recently. And the idea was, well, let's do proteomics on the SPI-2 secreted proteins and identify all the proteins that are secreted by this type 3 system. And so we do that. We basically use this proteomic differential labeling technique called SILAC, stable isotope labeled amino acids in cell culture, which is really just a fancy technology for growing your bacteria or your mammalian cells in two different media, one that labels the lysines and arginine with heavy isotopes and one that labels them with light. So you get two populations. The trick is to take these two populations, which in this case are going to be differing. This is a SPI-2 secretion system mutant, so there's no SPI-2 secreted proteins here. This is wild type. You then take the supernates in this case, and then you basically pool all these things and run them through mass spec. And because of the differences in the mass, you can then determine the masses of the various things. And if the ratio of the two masses are 1, basically that means it's secreted under equal conditions here and here. But if the ratios are different, and if they're higher, say in the wild type versus this one, it means there's more in this than this. That hints that maybe because this SPI-3 is knocked out, this SPI-2 type 3 is knocked out, maybe it's a SPI-2 secreted protein. So we did this, and it actually worked really, really well, because this is basically the list of all the proteins that have a high ratio being secreted in wild type compared to the type 3 mutant. And you can see we analyzed all these things here. And generally speaking, a ratio over 2 is kind of the noise cutoff level they use in these studies. And you can see tracking on here that here's one protein that's known to be secreted by the type 3 system, and one protein that's not known. This actually was ribosome protein here. And then all the known SPI-2 effectors. The neat thing is we identified all the known SPI-2 effectors that we'd found, and we found a whole bunch more that we then went on to actually study this. So as I said, this just came out, and I don't want to go into too much detail, and you can check the paper here, but here's the list of the proteins that are basically secreted preferentially by wild type compared to a SPI-2 mutant. And you can recognize many of the SPI-2 effectors in here, as well as others that we're now chasing down to try and confirm whether or not they're actually a SPI-2 dependent system. So what you find here is you then have to do additional studies to prove whether it's not secreted by SPI-2. Usually put a tag on it and try it in wild type versus SPI-2 mutant. So anyway, I think we're getting much closer to identifying what is the repertoire that Salmonella has in terms of SPI-2 effectors. And then having that, then the hard work begins, because now we've got to figure out what each one of these things does and how it affects the host cell. So in these days of systems biology, one is always thinking of ways of trying to figure out what the heck do all these things do. And Michelle Buchner is a grad student in the lab. She basically took all the known SPI-2 mutants before we just did that study and basically made a mutation in each one of these things. And the idea was to systematically compare them all at one time through a bunch of different screens. And the reason we wanted to do this is most of us in the field usually pick one effector, and then we work through what it does, but we never compare it to the others. And we felt that, well, we're going to have to bite the bullet and systematically define what do all these SPI-2 effectors do. So Michelle made all these mutants, and we started screening. One of the easy screens you can do is measure their replication inside cultured epithelial cells, HeLa, and I'm also not showing CACO2 cells. And based on what we knew, you'd predict that SPI-2 would not have an effect in Salmonella replication in these cells because they're epithelial cells, and we didn't expect to see any effect, and the bottom line is we didn't. So SPI-2 doesn't affect replication in epithelial cells. We already knew that because this is a SPI-2 mutant here compared to wild type, and they're the same. Okay. Well, what happens when you throw in macrophages? Well, there's where things heated up a bit. And so obviously, if you take a wild type versus a SPI-2 mutant, you can see the SPI-2 mutant is basically can't divide inside macrophages. So that argues that some of the things being secreted by SPI-2 are needed for replication in Salmonella. So you can see the starred ones here seem to have an effect. And based on a bunch of studies that Michelle had done, we've now been able to identify which effectors we think are key for replicating in these macrophages. Some were known, some weren't, and you can see them listed up here. So I think the moral of the story right here, you see, is that obviously several effectors are involved in this macrophage replication, but not all of them. So obviously these things probably have other roles, or we're just not seeing the effects inside cultured mouse macrophages, which you could argue might be a little artificial. Okay, what happens when you turn to the animal? Well, again, Michelle did this and looked in the spleen, the liver, the mesenteric lymph nodes. And first of all, you can see in all cases, when you compare the wild type for the SPI-2 mutant, SPI-2 mutants die. They basically can't live after 5 days inside these organs, and that's what we expected here. But what you see is there's some effects by some of the type 3 effectors, but many of them don't have pronounced effects in terms of long-term survival inside these animals. We expected to find these two here because it was reported in the literature that they were important, and sure enough they were. But really we couldn't come up with profound, severe phenotypes with many of these others. And that's not surprising because that's a theme we see a lot of the time with all these type 3 effectors. When they have repertoires of, say, 40-plus proteins, many of the times you can't see strong phenotypes for a number of reasons, perhaps redundancy, duplication, or the other reason is that they probably all work together. When you just knock one out, you haven't really disrupted the system enough to see an obvious phenotype. But anyway, that sort of gave us an overview of all the different SPI-2 effectors that we knew at that time and what they actually might contribute to disease. Okay, so now I want to turn to the concept of let's talk about Salmonella dog and what's known, and I want to show you that basically I think we kind of got it wrong a little bit, and maybe we need to rethink how we actually think about Salmonella. So when Salmonella comes in orally, it's acquired, it's ingested, it's then able to penetrate the intestinal epithelium. And I've just told you the invasion of these cells and everything is really dependent on the SPI-1 type 3 system. And we know that, there's data to show it and everything. Then the idea is that the infection then progresses a bit more. The bacterium goes systemic, gets in the reticular endothelial system, goes to the liver and the spleen, and for this time the SPI-2 system really kicks in. Everyone says, including us, that SPI-2 is later in this whole scheme of things. And that's, wherever you read textbooks, reviews will all tell you this. So Brian Coombs was a MD PhD student in the lab, and he was involved heavily in the early work when we were trying to set up a gastroenteritis type model in the mice. And he and others showed that if you pre-treated these mice with antibiotics, by shifting the microbiota, instead of getting a typhoid type disease, you actually get gastroenteritis or a diarrheal type disease in the animals. Very different disease. And so he was looking at this type of system, and he set this up. And one of the first things is, well, I wonder what happens in SPI-1 and SPI-2 mutants. Now, you predict SPI-1 mutants to be affected because this is a gastroenteritis disease, and that should have a profound effect, and that's what he saw. He also developed a pathology scoring system, I should add, that basically is a way of scoring disease, which was kind of a new addition to our lab, being a molecular biology lab, of actually scoring disease-type symptoms so we could actually measure disease. Well, what he found, first of all, he confirmed that if you compare wild-type versus a SPI-1 mutant. So this has no SPI-1 type 3 secretion. You can see this has severe disease, big swelling, fluid, infiltrate, etc. Compared to the SPI-1, this is what normal looks like. So basically it's attenuated. And that made sense. That's what the literature said. But what he noticed is that the SPI-2, this is not nearly as sick as the wild type. So if you knock out the SPI-2 system, remember this is early, this is diarrheal type things, we're seeing an effect of the SPI-2 system here. And you can also quantitate that histologically. Of course, wild type has a lot of disease. SPI-1 mutant doesn't really have much disease at all. But look how decreased SPI-2 is. In all cases, SPI-2 is quite a bit lower than the wild type. So that's weird because we didn't expect SPI-2 to have any effect in the gastroenteritis type aspects of this disease. And that was the first real warning light we had that SPI-2 may be doing something that we previously didn't appreciate. So to complement that, we started to look at the expression of SPI-2. And I've already told you SPI-2 should be on late. So we would expect to be on really late in the infection, days into the infection rather than early in the event. So Nat Brown was a postdoc in the lab at the time. And what he did is he set up a system of measuring when do promoters go on when you add these bacteria to animals. And this is something called Rivet, which is a technology that allows you to have a marker here flanked by these resolve sites. And then you take your promoter of interest and you clone a resolvase downstream of it. And if this promoter goes on, the resolvase comes on, it kicks this out of here, and then you lose this marker. So you can plate the loss of this marker. So this is a really good way of figuring out when these promoters go on. So the first thing we did was take a SPI-2 promoter and then add SPI-2 to macrophages, and we found that if it didn't go into macrophages, the SPI-2 gene was not on. When it went in, it resolved, and it then went on. So that makes sense. SPI-2 comes on deep inside cells later in the events. But what Nat did was then have a look at what happens actually in the animals. And we started 5 days into it, SPI-2 was on. Okay, that makes sense. 3 days, 2 days, 1 day, SPI-2 was on. Then we started to get desperate because we really didn't think it would be on that quickly, and that started marching backwards time-wise. And by 15 minutes, 80% of the SPI-2 promoter was on after we added the bacteria to these animals. And that was bizarre because SPI-2 should not be on that early in these various organs. So it comes on as fast as, basically, as soon as we can detect it, SPI-2 is already on in here. And that goes against everything we sort of had thought about SPI-2. And we also showed that SPI-2 regulator is involved in this resolution. If you knock this out, of course, it can't come on. And non-PARs and other regulators seem to have an effect. So that started to question, well, if it's on at 15 minutes, Salmonella is not even inside cells at 15 minutes. What's actually happening? So the lab then had a look at where Salmonella was at 15 minutes, and you can see the bacteria lined up here. So they're outside cells. Here's the intestinal epithelium here. And they all seem to be outside cells. Oops. Yet at this time, basically they're resolved. So then we even took a SPI-1 mutant, which says you can't get into cells, and looked at the promoter. And again, basically, even though they're not getting into cells, so this is an in-vein type SPI-1 knockout, they shouldn't get into cells, yet SPI-2 is on. And that was very different than what we expected in the whole SPI-2 regulation. So to confirm it, at the time, we turned to another animal model that's used to measure gastroenteritis, and that's a bovine loop model, and Brian Coombs involved in this. And up until the mouse model, this was really the only model for studying the role of Salmonella in gastroenteritis. These cows get gastroenteritis. You then just basically measure the fluid that accumulates in these loops. And of course, you've put in wild type, you get big fluid accumulation. And if you get a SPI-1 mutant, of course, you get a decrease in fluid. But look at this. We're seeing a decrease also in the fluid accumulation in a SPI-2 mutant. And that was very perplexing, but the data overwhelmingly basically always told us that SPI-2 played a big role in the fluid secretion in the early events of gastroenteritis. So based on all those studies, we've now compiled a lot of evidence to argue that we kind of need to rethink how we think SPI-2 works. And the concept that, no, it's just way late, long inside the organs and everything. And we know that SPI-2, whatever it's doing, whatever these effectors are doing, are actually kicking on very early, as soon as it's inside the intestine, even before it gets into cells. And it's getting ready to do whatever these SPI-2 things are doing. And it's needed for the whole gastroenteritis type aspects of the infection. Okay, I'm now going to really switch gears, and we're going to talk about Crohn's disease. And you're probably saying, this is a talk on Salmonella. Why are we talking about Crohn's disease, which is an inflammatory bowel disease? But hang in there, because I think you'll start to see what I'm getting at. So just to remind you, Crohn's disease, it's an inflammatory bowel disease. It's a really awful, severe intestinal inflammatory type disease. These are the lifetime risks of acquiring it. And a major complication of Crohn's, the most major complication of Crohn's, is they develop what's called fibrosis and strictures. Basically, the intestine gets really stiff and often has to be cut out by surgery. You can see the surgical removal of these parts of the bowel, and these can then redevelop these strictures. And this is just an awful aspect of Crohn's disease. So the intestinal fibrosis has been known for a long time. Maybe you can see the effects on the intestinal cell here. And basically, it's a huge deposition of collagen produced by these fibroblasts, myofibroblasts, that lay down all this collagen that causes this. And the problem with this is that it obviously causes severe things. But the other problem is, in this whole field, is there's virtually no mouse models. The only model is basically inject these chemicals into the intestine of these animals every day, and maybe once in a while you get some that develop these strictures, but it's very irreproducible. And really, studying this whole concept of this fibrosis has been very difficult in these in mouse models, and because of that, that's hindered the understanding of how this actually works. So going back to Salmonella, to try and tie these two things together, Guntram Grassel was a postdoc in the lab, and he decided, well, let's look at longer times of Salmonella, really longer times in terms of days and what's actually happening in the intestine. Now, in Salmonella, there's two kinds of mice: those that will die within a week, or those that we call resistant and live and don't die. So obviously, for these kinds of experiments, we have to use the resistant animals, those that have the NRAM gene that won't kill the Salmonella. And that way you can study long-term intestinal inflammation. So the idea is to use the gastrointestinal models we pre-treat with antibiotics, and then we're just going to follow over time what happens in the intestinal inflammation. And we're going to quantitate a whole bunch of different ways. And one thing we saw is that the Salmonella causes this chronic inflammation over a long time period, 40 days plus. One of the readouts of disease is basically to measure how shrunken the cecum is. The more shrunk it is, the more disease you have. So you can measure basically even just the weight of the cecum. Here's a nice normal healthy cecum. You can see these are very small and shriveled, and they have much decreased weight over time. That tells us this is sort of a chronic inflammatory type disease from the Salmonella point of view, and it lasts many, many days. Well, then it was Guntram and Yannette Valdez was in the lab. They noticed these really hard, funny things. And Bruce Valence, who's another colleague of ours, happened to be in the lab and said, "Have you ever stained for collagen?" And Yannette and Guntram really hadn't thought about this, but Bruce, working in the inflammatory bowel area, was already thinking of this. And so they then started to stain for collagen. And what they found is there's this massive depositions of collagen of these things, of low mag and high magnification. There's this huge collagen deposition. Looks very much like the strictures and the fibrosis structures you see in Crohn's disease. You can actually look at the upregulation of the collagen genes, and Salmonella, 21 days into it, causes a massive upregulation. It's hard to see here. Massive upregulation of collagen genes, and you can also measure the inflammatory cytokines. Let's get to here. And what we found is that this looks a heck of a lot like what you see during Crohn's disease. So you can measure various inflammatory cytokines 21 days after infection, and you can see the correlation in our model with those that are involved in inflammatory bowel disease. And the ones in reds are the ones we actually measured. Huge correlation. So it seems like we've sort of got a chronic intestinal inflammatory model that mimics Crohn's disease, including the intestinal fibrosis and the collagen deposition. Salmonella also induced the production of profibrotic cytokines. These are associated with triggering cells to produce collagen and deposition. And like we had hoped, we saw that. And we also saw cells that are involved in depositing collagen and fibrosis come down onto these things. And there's a huge, you can stain by this vimentin, which is a marker for these things. And these collagen deposition cells are found in large numbers in these chronic inflammatory intestinal areas. Salmonella, for its part, well, that's hard to see, is basically it's found, you can see the Salmonella here, it's found really in the lumen of these things. We don't see it deep intestinally. It seems to be sort of on the surface where it's triggering this inflammation. We don't see it deep in the tissues. It seems to be mainly in the lumen in these infected areas. But it's kind of like this, piece of sand in the oyster causing this intense inflammation and irritation in this, triggering these whole events. We had a look at what the type 3 systems are, and we found that both SPI-1 and SPI-2 are needed for this. And if you have mutants in both SPI-1 and SPI-2, you have less type effects in here. So they contribute to this whole process. That makes sense because you need this long-term intestinal inflammation happening. Now, if we're going to develop a good model for fibrosis, you have to use it in mice that people can make knockouts in. And so we, and most of them use a type of mouse that's NRAM sensitive. So we then developed the model using basically a slightly attenuated Salmonella. This is a live attenuated Salmonella that doesn't kill mice. It's also used as a vaccine strain. And if you use this strain in these mice, and the mice don't die, you still see the intense inflammatory type effects going with this. And you get large collagen deposition. And what this has now done is allowed this model to be extended to various knockout animals to start to probe the role of various inflammatory pathways, et cetera, in this type of disease. So this has actually turned out to be really useful from the gastrointestinal point of view because they really didn't have a good mouse model for Crohn's until they've come along. And the gastrointestinal people have embraced this, although I get all these funny phone calls from gastroenterologists like, how do I grow Salmonella? You mean you want me to infect animals to do this? But we give them the airway vaccine strain, so it's a vaccine strain, so they're not going to hurt themselves doing it. But it's now being embraced by many labs, and it turns out it's actually a really, really nice system to study fibrosis, which is the big, big problem in Crohn's. what it has to do with Salmonella pathogenesis. It's a good model to study chronic inflammation, although there's no literature that we can find on Salmonella triggering fibrosis in humans doing long-term inflammation. Okay, so we talked about the SPI-1, the SPI-2, there's effects and all the effectors they're having in cells. I now want to talk about an area that is a hallmark of Salmonella typhoid, but really hasn't received much research attention. And this goes all the way back to Mary Malone, also known as Typhoid Mary. She was this Irish cook in New York, and she basically shed Salmonella typhi from her gallbladder into her feces. And one wonders about her cooking ability, but that then led to the contamination of other people, and a lot of people died of typhoid fever because of her. She was incarcerated twice, and basically the second time they locked her up and put her on an island in New York for the rest of her life. And she refused to have her gallbladder removed, and she basically shed transiently her whole life. They were very hard on her. You can see they depicted her. Back in this era, women didn't usually have hairy faces. They didn't usually have double chins. They didn't have hair in their arms. And so this was actually a newspaper editorial cartoon of this woman doing this. She swore she'd never have typhoid fever, and it's a very interesting story in terms of public health measures. The first time people were locked up for public health reasons. But anyway, she sheds typhi. And the question is, well, how does she shed typhi? Well, it's shed out of the gallbladder. And the gallbladder is basically between the liver and the intestine. It's where bile salts are concentrated. And it's really a little sac. There's a cross-section through it here that's full of bile. And then when you eat, etc., bile is delivered down the bile duct into the intestine. And it then basically aids in the digestion. So it's a little sac that stores bile. It's also thought to be where Salmonella can hang out and basically cause this carrier status. So in terms of what's known with gallbladder and Salmonella, it's known that Salmonella typhi, patients can have Salmonella typhi in their gallbladder, both acute and chronic type patients. It's well known in the gallbladders of rabbits in the early 1900s that the bacteria can persist in the gallbladder. And Denise Monack showed a few years ago that sometimes you can test the gallbladder of mice in her persistence model. And there's reports in literature that there's changes in the gallbladder during typhoid fever. A lot of times there's basically disease in the gallbladder. And it's thought that this sort of serves as the way of seeding the bacteria into from the liver into the bile and then causing shedding. But the problem was that really we didn't know anything about it. We had this literature telling us these things, but we really didn't know. There was no data on Salmonella infections of the gallbladder and how it worked, what it might be doing. So the idea was, well, let's see if we can't set up a model. This is set up by Ellen Arena, who's now in Philippe's lab, and Alfredo Menendez, who was a postdoc in the lab at the time. And the idea is to take mice and really give them infections, then we'll just collect tissues at various time points. I started this just as a project to see where Salmonella was going in the mice at all organs, including gallbladder. And they harvested these things and then just had to look at where the bacteria counts were. And when you basically fractionate a mouse and you look where Salmonella counts come, in the early times, 6 hours after infection, they're mainly in the liver and the spleen and the kidney, and there's a little bit of a dip, and then there's a log scale here. You can see they go up and up and up in the liver and the spleen until 120 hours into these things. Well, when you start looking at the gallbladder, you start to see a few bacteria by 48 hours, but by 120 hours, the number of bacteria per gram of tissue is basically greater than that found in the liver and the spleen. There's a lot of Salmonella in these gallbladders. And so that then led Ellen and Alfredo to think, well, maybe there's something to be had here, and let's have a look at what actually goes on in the gallbladder. So when they started to look at these things, you could see Salmonella both inside cells here, and also extracellular bacteria. You'd expect to see them given the high numbers of Salmonella that are present in this organ. And again, just to remind you, this is what a normal healthy gallbladder looks like. You can see nice fingers pointing out. And this is basically filled with bile. When you infect with Salmonella, all heck breaks loose, and these poor gallbladders get extremely diseased. And you can see here's 4 gallbladders in 4 different animals showing severe infiltrate and also very destruction of the whole structure of these things. And large disease, infiltrate, etc. here. So when they started to look closely where Salmonella is, they found it didn't translocate in the deep tissue. It seems to be on the mucosal surface. And it's hard to see here, but this is the apical surface of the cells. And down at the basolateral surface, they started to see a lot of the Salmonella. But they were just hanging there. They weren't going penetrating deeper than this. Now, we started to look, well, are they inside cells and they localize with various markers? And LAMP1 is a marker that stains lysosomal glycoproteins. And so normally there's a nucleus here, and the LAMP1's up in the apical surface here. When you affect with Salmonella, all the LAMP1 up here disappears, and it all traffics to the Salmonella, which are at the basolateral surface of these things. So there's a big rearrangement in the vesicular structures in these cells, and LAMP1 then targets to the Salmonella. And this is a big indication, probably, that the Salmonella are inside cells in these LAMP1 positive areas. And here's another section that Ellen showed. You can see all the Salmonella just all along the edges here, basically at the basolateral surface of these cells. And you can see it more here. You can see the green is the actin. You can see the apical surface up here. Look at all the Salmonella all along the basolateral surface of these things, basically in the lumen of the gallbladder, but the opposite end. And the reason this is weird is that in intestinal epithelium, in the intestine, Salmonella is always at the apical surface. And there's no other cell types being reported where Salmonella hangs out at the opposite end of these cells at the basolateral surface. And then with Wayne Vogel, we had a look electron microscopically what's going on. And again, you can see the bacteria are way down. Here's the apical surface, way down at the basolateral surface. Lots of bacteria inside vacuoles, probably LAMP-containing vacuoles, and even replicating inside vacuoles here, down at the bottom of these cells. And they seem to persist here. And this is where they then replicate and live inside these cells. They seem to replicate. You can find pictures. The bacteria seem to be dividing in these vacuoles, and much like you see in other epithelial cells, except, as I emphasize, they're underneath, they're the basolateral surface instead of the apical surface where they normally in other epithelial cells hang out. So a question we had is, can Salmonella grow in bile itself? Bile is a salt. It's basically a detergent, and it's thought to be, degrades food, and there's ideas that degrade bacteria, etc. So they had a look, can Salmonella grow in concentrated bile? So they got hold of some bile, and they then basically added it as a growth media. And what they found is that Salmonella grew just fine inside bile, as well as in L broth, and it grows just fine here. And Salmonella doesn't grow very well in PBS. So not only does it survive inside bile, it actually uses as a growth medium to live inside these gallbladders. So bile is definitely not antibacterial for Salmonella. So to put the whole gallbladder story together is that we know the gallbladder is heavily colonized by Salmonella. It seems to go to the basolateral surface and hang out there. You can see the other things here. And it seems to trigger major infiltration of neutrophils in there. And so it seems to be a really neat but different system to study Salmonella biology, because all of us until now have been really studying Salmonella either in macrophages or in intestinal epithelial cells during the gastrointestinal type phases. And so this opens up studies in the area of carriage and dissemination, and then which obviously plays a major role in the Salmonella Typhi biology of the disease. So one of the things we noticed is that Salmonella seemed in the gallbladder to cause the accumulation of things called lipid droplets. These are these little things here. There's one here and one here, a few more around in here. When you infect with Salmonella compared to the uninfected one. And that led Ellen and Sigu Otware in the lab to then start to have a closer look at this. This is a really rather bizarre phenotype. So if you're like me, the first thing you say is, what is a lipid droplet? I didn't know what a lipid droplet was, but luckily Ellen and Sigu told me. And basically these are intracellular stores for neutral lipids, and they sort of balls are formed. We put the lipids in the middle, you surround them by a phospholipid bilayer around the outside, and you then accumulate these things, and you can then basically keep these things inside cells. They also had a look to see what happens in their suggested role in pathogenesis, because we were seeing them in the Salmonella-affected cells and not other cells. And there's some hints in the literature of some organisms involved in this, but there really wasn't very much. So I'm now going to tell you the story of the identification of Salmonella SPI-2 effector that basically seems to be involved in triggering this lipid formation. And to do that, I have to go back to our proteomics screen, the SILAC technology. Again, remember that's that heavy and light labeling. But instead of labeling bacteria this time, we're labeling tissue culture cells. So mammalian cells are being labeled. And in one basically extract mammalian cells, we've got a tagged SPI-2 effector. As you'll see, it's a molecule called SseL, for example. And the other basically is just the standard HA tag alone. And the idea is to immunoprecipitate with this tagged effector versus the tag of this light and heavy type media, pool the samples, do mass spec again, and look for those that have differential labeling ratios that are very different than the standard ratio of 1. So Siggy and Amit were doing this, and the bottom line is we did this on pretty much all the SPI-2 effectors. The idea was going to find binding partners for them. I'm not going to go through all of them, but the studies just now come out where we, first of all, we confirmed all the known SPI-2 effectors, what they bind to in mammalian cells of the ones that were known. So that was good. It means the technology works. Plus, we found some new ones, and you can see some of them listed here. It was a bit disappointing because many we couldn't identify binding partners to. But for the ones we did find partners, we seem to be able to confirm these things, and they seem to be real. So we're happy with that. So the one we focused on is this one here, just for the sake of this story, because I'm telling you about these lipid droplets. This is a SPI-2 effector called SseL. And it turns out it bound this protein called oxysterol-binding protein, OSBP. This is a host protein, and it's thought to bind oxysterols, so that might be a clue in terms of lipid. It's involved in vesicular trafficking, maybe that involves lipids, sphingomyelin synthesis, cholesterol metabolism, all things that might be involved in lipid droplet formation. In terms of SseL, we knew it was a SPI-2 effector. The mutant was mildly attenuated, and others had shown that had this deubiquitinase activity. Basically, it removes ubiquitin from various things, although the substrate of whatever gets deubiquitinated was really not known. Okay, so SPI-2 effector binds this OSBP. What does that do? Well, the first thing Siggy did was to prove this interaction was real. We showed it once on proteomics. Again, I don't want to go through the details of this plot, but bottom line is that if you pull down with OSBP, you pull down the tagged SseL. And if you do the reciprocal experiment, pull down SseL, you pull down OSBP. So that confirmed the proteomics. These things do tend to bind, and proteomics was telling us, well, we think it was telling us the right thing. So then what happens? Where does OSBP go? Once we had a look where we couldn't see it in these cells, and we basically started staining for OSBP, and it's a little bit hard to see, but here's Salmonella here. You can see this OSBP label seems to co-localize with Salmonella. It's really hard to see up here, but on the screen it's quite strong. You can see OSBP seems to be co-localizing in the area of Salmonella, again here versus here. So, actually, maybe you'll see it better on the merges up here. So there was co-localization, so that's kind of guilt by proximity, the fact that OSBP is co-localized near where Salmonella does. This is just more of the same, just again proving that bacteria seems to co-localize with OSBP. And this is in gallbladder, so that's good. It means it's co-localizing. So, okay, if SseL is affecting things and OSBP is needed, the odds are that if we basically downregulate OSBP in the host cell, maybe that'll affect Salmonella replication. So Siggy used siRNA. In this case, basically wild-type scrambled siRNA, basically Salmonella replicates like this. And then when you knock down OSBP, replication goes down. So that argues that OSBP is involved in it. And if you transfect in more OSBP into cells, you get an increase in Salmonella replication. Okay, that's all shaping up nicely. Well, we did one experiment too many. It's always the case in science. So, okay, if SseL is needed for it, you predict that SseL, if you took a mutant, that should disrupt the whole thing. And this is where the wheels fell off, unfortunately, and for whatever reason, that when we mutated SseL, which was binding OSBP, we thought it would alleviate the effect. It didn't. Salmonella still was decreased with the OSBP, and there's still, when you transfect, increased more. So it's more complex, as always in science, than originally thought, and that still is a bit confusing. Okay, let's go back to the gallbladder and see what's going on with this SPI-2 mutant. So SseL, when you take a mutant, one of the things we saw why it's hard to see, is that Salmonella is really elongated in these, it's hard to see, but there's really long red things in here. In the SSEL mutant, there's some phenotype going on in terms of Salmonella shape. But what's really neat, and let's get back to lipid droplets, when you take a mutation SSEL, you got a gazillion lipid droplets. Look at all these things here. So here's wild type. You can see a few lipid droplets. There's none in uninfected cells in the gallbladder. But look at all the lipid droplets in here. Phenomenal amounts of lipid droplet formation in here when you knock out the SSEL. So it seems to be that SSEL has a function kind of suppressing formation of too many of these lipid droplets, and when you knock it out, you get way more than you do in the wild type. And you can see some spectacular events in the SSEL mutant. So here's uninfected gallbladders, wild type infected, has a few lipid droplets, but look what happens in the SSEL mutant. Profound effects in the cell. You can also stain the lipid droplets with this thing called Bodipi, which is a lipid stain, and you can see that in the mutant you get a lot more of these lipid droplet staining here. You can even extract the lipids in these things and just measure them, and you can see that in the SSEL mutant there's just way more. You can just see these things here. There's just a lot more lipids in here, and you can measure the lipids being produced here, and you can see there's huge amounts of lipids in this SSEL mutant. So whatever's going on, SSEL seems to be suppressing the formation of these lipid droplets, and when you take out SSEL, still infected with Salmonella, the gallbladders just go crazy in terms of lipid droplet formation. So if that's the case, now I told you that it was known that SSEL had deubiquitinase activity, so it basically gets rid of ubiquitin. So we basically put a point mutation in this activity and see if that had any effect on the cells. And it turns out that, that the lipid droplet accumulation depends on the deubiquitinase activity. And you can see when we took an SSEL mutant to put in wild-type SSEL, we suppressed lipid droplet formation. Yet if you put in a point mutant in the deubiquitinase, you still get a lot of these lipid droplets here. So that argues that this enzymatic activity of whatever it's deubiquitinating is key in this lipid droplet formation. And this is also shown here with this Bodipi stain. Here's Salmonella SSEL mutant with a wild-type SSEL. You get a few lipid droplets here, but if you take the point mutant, you get a lot more, meaning you need the deubiquitinase to suppress the lipid droplet formation. So that kind of brings me to the end of what I want to tell you about Salmonella. We've gone through many different aspects of it, but what I wanted to do is sort of bring up some of the areas that are different from the way we think about it and what these effects are and how they contribute to Salmonella. So one of the things I try to drive home is depending on where Salmonella is inside the intestinal tract or in the liver and the spleen, you get very, very different diseases. And that argues you've probably got different virulence factors and different bacterial factors doing this. In mice, normal mice, you get the systemic type disease, and I've talked before while I've been over here this month on the effect of antibiotics in the microbiota. If you change the microbiota, you get a whole different disease. You get gastroenteritis. I showed you some data that the long-term infection of Salmonella can cause this fibrosis type effect, and this has now turned out to be a very nice model for studying fibrosis as a model of complication of Crohn's disease, as well as just studying long-term inflammatory type infections. I spent a fair bit of time telling you about this gallbladder because I think this is an important aspect that really hadn't been studied before about how it contributes to the whole shedding of Typhi. And the neat thing is it gives you a whole different way of looking at how Salmonella interacts with different epithelial cells, being basolateral instead of apical. And we know that the life in the gallbladder plays a key role in the whole shedding and dissemination of typhoid fever. And now we're starting to get actual answers of how it hangs out inside the gallbladder and what it might be doing. So there's no doubt Salmonella interactions with the host are complex. There's an awful lot of these effectors doing an awful lot of different things. And unfortunately, there's often an awful lot of things we just don't know yet. And so, we anticipate the years to come. We'll have lots more things to do because we think we're getting close to the end of identifying new type 3 effectors. We think we've got the list pretty much there. But each effector really takes a whole, as I showed you with SSEL, takes a whole bunch of effort to try and track down what that one particular molecule is doing inside these cells. And so there's lots of things we have to do yet in terms of understanding really at a molecular level how Salmonella interfaces with its host. So this is a picture of the lab, one of the retreats. And obviously I've mentioned many people's work. I think I can detail them here a little bit more, and the various people involved in the SPI-2 work, the early SPI-2 work in the gastrointestine. Guntram heavily involved in the fibrosis model, and the gallbladder, Alan Alfredo, and some other friends were involved in this. And then Michelle did all the SPI-2 mutants, and Siggy and Ellen have done the whole SPI-2 effectors and also the whole effect of the effect on the lipid droplets. And for those of you who haven't been to Vancouver, this is Vancouver. It's on the west coast of Canada. UBC, the university, is actually out on the peninsula right here. This is where the university is. This is the downtown area. And, it's just out of the picture where the Stanley Cup finals are going to be played. There's an arena right here. And there's a nice park here, et cetera. And this is Vancouver Island up here. So with that, I say thank you very much.